Today, we're going to talk about a life form that can literally survive the vacuum of space and a dose of radiation that would kill you or me 1,000 times over. And we're going to find the tiny molecular switch that lets it reboot after a total catastrophe.
I mean, it sounds like something straight out of science fiction, right? But some organisms have already figured it out. So how do they do it? How can any living thing withstand forces that would just annihilate almost everything else on this planet?
Okay, let's dive in. I want you to meet Deinococcus radiodurans. This little bacterium is just in a league of its own. It can take a radiation hit over 1,000 times what would be lethal for us. It can completely dry out, lose all its water, and then just come back to life. And get this, it actually survived for a full year attached to the outside of the International Space Station. We're talking the vacuum, the cold, the radiation of space. Unbelievable.
So to really get how it pulls this off, we first have to understand the absolutely catastrophic problem it's solving. We need to look at what happens to its genetic blueprint when it gets blasted with that much radiation.
You can think of radiation as a high-speed molecular shredder. As it tears through a cell, it just pulverizes the most important molecule inside, its DNA. For Deinococcus, we're not talking about a little bit of damage. Its entire genome is shattered into hundreds of tiny random fragments.
Now, we're talking about a very specific and very catastrophic type of damage. It's called a DNA double-strand break, or DSB for short. This is where both backbones of that DNA helix are just completely snapped in two. For you, me, and pretty much all other life on Earth, even just a few of these breaks, and it's game over. The cell dies.
But Deinococcus, it has a totally different game plan. It doesn't just survive this. It practically thrives on it. And the reason it can do that is because it has this unique damage control system, all controlled by one very special molecular switch.
You know, most bacteria, they rely on the standard, well-known system called the SOS response to deal with DNA damage. But Deinococcus, it doesn't even have it. It's like it threw out the standard playbook and evolved its own, way more sophisticated system to manage the crisis. And the central coordinator, the real hero of this whole unique response, is a protein called RqkA. It's a special kind of enzyme known as a protein kinase. And honestly, the best way to think about RqkA is as a biological master switch. When total disaster strikes, RqkA is the switch that gets flipped to on.
Okay, so how does this switch actually work on a molecular level? Well, the process is called phosphorylation. Here's how it goes down. First, the radiation shatters the DNA. That's the signal. Second, RqkA senses this chaos and switches on. Third, it immediately seeks out its main target, which is another protein involved in DNA repair called PprA. And fourth, and this is the key action, it attaches a tiny little molecule, a phosphate group, right onto PprA. This one tiny chemical tag completely changes how PprA behaves.
So let's zoom in on that target. What exactly does adding this phosphate group do to PprA? And why is that so incredibly important for putting a completely shredded genome back together?
All right, so PprA is a crucial member of the DNA repair crew. You can think of it as a first responder. Its main job is to find all those broken, frayed ends of the DNA fragments and just grab onto them, holding the pieces of the puzzle in place for the next stage of repair.
Now this slide shows the effect of that master switch perfectly. Before RqkA flips the switch, PprA is in its, let's call it, default mode. I mean, it can bind to DNA, but it's not super efficient. But after RqkA tags it with that phosphate, it enters supercharged mode. It becomes this high-efficiency DNA binding machine. And here's the number that really tells the story. Research shows that after RqkA tags it, PprA has a 4-fold higher affinity for those broken DNA ends. It becomes 4 times stickier, grabbing onto the damaged sites with just incredible efficiency.
And hey, you don't just have to take my word for it. This right here is the direct evidence from the lab. What you're looking at is called a gel. On the left, you've got the regular, untagged PprA. You can see it starts binding DNA as you add more of it. But just look at the right side. That's the phosphorylated, supercharged version. It latches onto the DNA so much more aggressively, even at way lower concentrations. Those darker bands up top, that's a protein that is super sticky for DNA.
So a stickier protein is a fantastic start. But the real test, the ultimate question, is whether this molecular upgrade actually translates into faster, more effective repair of the entire shattered genome.
And this is the exact question the researchers asked.
I mean, it's cool that it has a 4x increase in binding, but does that really make a difference when you have to stitch hundreds of DNA fragments back together perfectly?
And the answer was a resounding yes. This slide is showing an experiment that tests DNA ligation. That's the actual stitching process. See those bright bands up top labeled IMLP? Those represent big, long, successfully stitched together DNA molecules. And with the supercharged PprA, you see this massive boost in stitching activity. It acts like a molecular matchmaker, efficiently bringing the broken ends together so other enzymes can come in and finish the job.
But here is the ultimate proof. This is at the level of the living, breathing organism. This graph is plotting survival against bigger and bigger doses of radiation. The top lines, that's our normal Deinococcus, just shrugging off the damage like it's nothing. Now look at the bottom lines. These are bacteria where that RqkA master switch has been broken. They're hypersensitive. They die off almost as fast as a normal bug would. This is the smoking gun, the direct link from the RqkA switch to the supercharged PprA protein, and finally to this bacterium's just unbelievable ability to survive.
So, why does all this matter? Why should we care so much about studying one incredibly tough little microbe? What can we possibly learn from its personal guide to surviving the apocalypse?
Well, understanding these hyper-efficient repair mechanisms gives us an incredible model to study our own DNA repair, which has huge implications for things like aging and cancer research. You can even imagine engineering organisms like this for bioremediation, to go in and clean up radioactive waste sites. But honestly, most importantly, studying this organism just fundamentally challenges our ideas about the absolute limits of life.
And that really brings us to the final thought here. Studying this organism isn't just about one weird bacterium. It's about dissecting a blueprint for resilience that challenges our most basic assumptions about what life can endure and how it can rebuild itself from the absolute brink of annihilation.